In this experiment, I'm testing how variables like acidity and food availability affect the growth of bacteria colonies inside an environment. So with our samples here, we have regular H2O, 3 milliliters, H2O with 2.5 grams of sugar, and that's also in 3 milliliters, but then there's the 2.5 grams of sugar added. And then we have H2O with HCl at pH of 1, H2O and sugar, 2.5 grams, and HCl of pH of 1. And then we have Coca-Cola, 3 milliliters, 2.5 pH. And what we're trying to see here is whether what type of effect each variable has and which one is the most detrimental to bacterial growth. So we'll see whether bacteria can survive in the H2O or any of the others, and then we'll see which sample does the best. To mimic the Coke, which also has low pH and high sugar content, we had the H2O and HCl and sugar. But in case there's something extra in Coke that allows for bacteria to grow or not, we'll know because we'll be comparing these two especially. Here we had BHI solution, which we used to take non-virulent bacteria, Listeria, and we put that inside 3 milliliters of BHI solution. That's getting ready, and those bacteria are growing, and in an hour they'll be ready. Here's some of the supplies, plenty of water bottle samples, Coca-Cola, sugar, all the tools. I've done this myself. I didn't get information off the internet. Weighed the sugar over there, and now we're gonna go make some plates so we can plate the bacteria when they're done growing. So I'm making the BHI and getting this ready. I added BHI into some distilled water. Going to make 500 milliliters, and then right now, as you can see, it's not very homogeneous, but we're going to take care of that. With magnets. Okay, let's turn it up. Yep, it's like a toilet. I'm going to be back when this is done. As you can see, we have a little bit of bacteria here. It's only grown for about half an hour, but that's enough. We're going to pipette 400 microliters of this stuff into each and every single one of these samples, then let them culture overnight and plate them tomorrow. So I just added 400 microliters of Listeria to the Coca-Cola. Now I'm going to do the rest with each one. Here's the H2O. This is a control. And that's done. Now these are going to culture overnight, like I said. We're going to put them in the shaker at 220 RPM, and they'll be in there at 37 degrees Celsius. And tomorrow we're going to come back and they should be cultured. And we're just going to, after that we're going to have the plates ready. Well, not ready. We're going to autoclave the plates because we're going to put everything in the fridge tonight. And then on Sunday, the last day, we'll count colonies, and hopefully almost all of these will have no colonies or just very few, because I don't want to count like 780 colonies for 6 plates, you know? It's going to suck. And because I have to write all this too, it's homework, you know? Can't just be here all day. Last one, this is H2O, sugar, HCl. Like I said, this is an important one because it's supposed to mimic the Coca-Cola, because that's what inspired this experiment. And if this has a varying degree of bacteria to the Coca-Cola, then obviously there's something in the Coca-Cola that's not in this simple solution that's causing that difference. So we have these guys ready now. We're going to take them and shake them. Voila, it's done. So after it was done centrifuging, after it was done spinning, I took out the magnet, I poured it in, added 7.5 grams of agar, because that's what it calls for, and the agar is going to make this stuff solidify very quickly. It's liquid now, but in a few minutes it won't be, so I'm just going to leave it somewhere. I gotta label it. I'm going to leave it somewhere. And tomorrow, like I said, we're gonna autoclave this stuff and then make it into plates, and then we're going to we're gonna have 6 plates total. We're gonna have H2O, H2O sugar, H2O HCl, H2O sugar, HCl, Coke, and the control listeria that's in BHI and frozen overnight. Well, not frozen, fridged overnight, so there's not a lot of growth. And that's it so far. See you tomorrow. The cultures are done. The plates are done. We're about to plate them and see how they grow. Okay, that took a while, but we managed. I'm going to go over what we have on the plates before we put these things away. We have H2O sugar HCl, that's 1x. H2O HCl, that's 1x. H2O and sugar, 1x. Now, I suspected I couldn't suspect how much bacteria was in the Coke because it was darker, so we just had 10x and 1x. So there's a 10x dilution and a pure Coke that we originally had. And the reason these only have 1x is because they didn't appear to have very much bacteria, but we don't know for sure yet until we actually grow them. And then this is negative control. This is the bacteria that we froze, that we put in the fridge yesterday, that we used to infect the other samples. These were not allowed to grow, but we made dilution so we could compare them to the H2O, which appeared to have the most bacteria. And it's not suspected that the H2O grew anything, but it is suspected that they're still alive, so maybe everything else here just killed it. Who knows? We'll see when we have the plates. We have 10 to the 5th dilution of the negative control, 10 to the 4th, 10 to the 3rd, 10 to the second 10x, and the pure negative control. And comparable to that, we have the H2O, which appeared to have the most bacteria. Let me see if I can, it's, I don't know, it's kind of cloudier than the others, and it was clear. Like, let me compare it to, let me compare this to something. Yeah, here's the H2O, HCl, right. On the left we have H2O, and on the right H2O, HCl. Obviously this one is going to have more bacteria, the H2O. Right. And so we have the H2O in 10 pure, 10, 100, 1,000, 10,000, and 100,000 dilutions. We're gonna put these at 37 degrees Celsius overnight, and we're gonna compare them and count bacteria tomorrow morning. Okay, we'll see you then. Now that we've analyzed the plates, we're going to take samples from the beakers that we've made, and now we're going to put them in some cuvettes and measure the DNA that's inside all of them to make sure we know which ones were most successful in growing bacteria. So we've gone through a couple samples. I'm writing down the results, and so far the H2O is well in the lead. Of course HCl didn't do so well. I'm measuring the OD 600, by the way, and the BHI was less than the H2O, and the sugar was actually the H2O sugar was actually very little, and I'll mention why that happened later on. Sugar. Okay, so I'm going to try to do this with one hand now. You open the lid. You have to grab it at the sides because the sides are rigid, and it has to be clear for the spectrophotometer to read it. Right. Gotta fix this here. I'll be back when I'm done fixing this.